Hello everyone, I am Rio Zefani Antonio and I will be discussing the role of cell competition in aging. Here are my table of contents: 1. Introduction, 2. Modes, 3. Results, 4. Summary, and 5. Conclusion. So without further ado, let's start with the introduction. Recent advances in rapid medical detection and diagnostic technology have extended both human health and life expectancy. However, aging remains one of the critical risk factors in contributing to major incapacitating and fatal conditions, including cancer and neurodegeneration. Therefore, it is vital to study how aging attributes to endangering human health via infliction of age-related diseases and what must be done to tackle this intractable process. So what is cell competition? It is the differences in the fitness status of cells within a population. It selects fit cells to populate a tissue at the expense of less fit or suboptimal cells that undergo elimination. What is fascinating about CC is the panoply of situations where it applies, one of which is aging. Aging can be defined as the process in which there is a progressive time-dependent decline of bodily function. While we get old, our cells do not age all at the same time, and this results in imbalances in the fitness status. Thus, it is vital for healthy aging that damaged or suboptimal cells are eliminated to promote tissue's well-being. Aging is one major challenge that almost every creature encounters. However, owing to improvements in medicine and healthcare condition, life expectancy has increased, meaning that it has advanced the health of the aging population. Let me add some information about cell competition. Cell competition was first proposed by Morata and Ripoll in 1975, using flies with mutation in ribosomal protein named minute mutants. When minute slow proliferating cells are in contact with wild-type cells, they are eliminated from the wing imaginal disc. This cell elimination requires local interaction between population with different growth rate within the same tissue compartment. Moreover, cells overexpressing DMYC are fast proliferating and outcompete WT cells in the wing imaginal disc in a phenomenon described as supercompetition. The discovery of supercompetition shed new light on the importance of CC, since it could be related not only to development but also to cancer. Now, let's move on to the modes of cell competition. At the present day, there are three modes of CC generally accepted, but only the two of them are detailed here, which is the fitness sensing and mechanical cell competition. Under fitness sensing, there are two subcategories: the flower fitness sensing and ligand receptor models. Then second mode is the mechanical cell competition. For the fitness sensing cell competition, flower was first described in Drosophila as a transmembrane protein with three different isoforms alternative splice, as shown in the figure 1a. Flower fitness sensing, in which looser cells will express flower loose, and if they are supposed to be eliminated, they also express azote and Hyd. So azote is another mediator of cell competition that acts as a fitness checkpoint, which consequently activates the pro-apoptotic gene Hyd, leading to cell elimination and delayed aging. Then for the second subcategory, ligand receptor models, wherein there is recognition of the ligand SLIT, SPATZO, and SAS by the respective receptors ROBO, TOL, and PTP10D, leading to the elimination of the looser cells. For the second mode of cell competition, we have the mechanical cell competition. Mechanical cell competition occurs due to the mechanical forces within a tissue that produce compressive forces, leading to looser cell elimination. Winner cells are represented in pale brown and looser cells in dark brown. black arrows represent the direction of the compression forces. In addition, recent literature has brought attention to a novel mode of CC, mechanical cell competition, in which mechanical forces subjected in a tissue lead to loser cell elimination. Now, we have cell senescence. This is when cells are not exactly unfit. In fact, they are often capable of normal function, but they are not operating for various reasons, and this happens more and more with age. In a simplistic definition, senescence is a stable and long-term state of cell cycle arrest, wherein the cells are still viable and metabolically active. Senescent cells generally do not contribute to the tissue well-functioning and can secrete toxic factors that can affect the neighboring non-senescent cells. Thus, removal of those cells is beneficial to the organism, and it is proved to ameliorate or revert diseases such as arthritis, AD, and cancer. Moreover, this will be further explained on the results. Now, we're up to the results of this study. The results are only highlighted on the dietary restriction, stem cell dynamics, and cell senescence. Let us first discuss the results of cell competition in dietary restriction. Using D. melanogaster, it is proved that dietary restriction slows the age-related decline in intestinal integrity by enhancing enterocyte cellular fitness through upregulation of DMYC in the intestinal epithelium. Reduction of DMYC in enterocytes induce cell death, which leads to increased gut permeability and reduced lifespan upon DR. Furthermore, reduced expression of MYC also extends lifespan and slows the onset of age-related pathologies like osteoporosis, cardiac fibrosis, and immunosenescence in mice, suggesting conserved effects of MYC on aging. Interestingly, DR has been pointed out as the most effective way to slow down aging. Cell elimination leads to punctual disruption of the gut epithelia, which increases its permeabilization, favoring the appearance of infections and reducing lifespan. By increasing the fitness status of the gut epithelia, CC is blocked, gut integrity is maintained, and the lifespan extended. On the contrary, a rich diet reduces DMYC expression, leading to CC-dependent elimination, and a decrease in the lifespan as explained above. It is interesting to notice that a slight upregulation of DMYC is beneficial. However, a high overexpression can even be detrimental. Next is the results on stem cell dynamics. Up to date, there is only one report highlighting the importance of CC in the selection of the fetal stem cells during aging, as shown in the figure 3b. Epidermal stem cells express collagen 17. Stem cells with high levels of collagen 17 divide symmetrically in horizontal axis and outcompete cells with low levels of collagen 17, which divide asymmetrically and are perpendicularly extruded by differentiation. Collagen 17 is downregulated in aged skin, which impairs the success of the selection of fetal cells, causing depigmentation. Lastly, the results in cell senescence. One gene involved in cellular senescence is the p53. When irradiated, hematopoietic stem and progenitor cells are mixed with non-irradiated cells, high-activity p53 cells are outcompeted and enter in p16-dependent senescence, assured in the figure 3c. It is surprising that the damaged cells in this context are not eliminated by apoptosis, but are maintained in stock. The authors suggest that by marking loser cells as senescent, They are gradually eliminated from the tissue if there are undamaged cells available for the replacement. To understand more the results, here is the summary of situations wherein cell competition plays a role. For the dietary restriction, there is upregulation of MYC in the enterocyte, reducing cell elimination, decreasing gut permeability, decreasing the risk of infection, and increasing the lifespan. In a normal diet, unfit cells with reduced level of MYC are eliminated by apoptosis, increasing gut permeability, increasing the risk of infection, and decreasing lifespan. For the stem cell dynamics, in young skin, epidermal stem cells with high levels of collagen 17 eliminate cells with low levels of collagen 17 by upward differentiation. Old skin is mainly composed of EPSC with low levels of collagen 17, since its expression decreases during aging. The arrow represents the mechanical forces exerted over the looser cells. Dash arrow represents the direction of differentiation. Then for the cell senescence, DNA damage, either by irradiation or aging, increases the level of P53 in hematopoietic stem cells, represented in red. HSCs with high level of P53 are outcompeted by lower P53 HSCs, which are the yellow, and enter in senescence, which are the pale red. In conclusion, the bottom line of this review is that the applications of cell competition are still being explored, but the outlook is very promising, particularly in the area of aging. To be clear, this research is not looking for a Benjamin Button scenario. There will not be a pill or a vaccine that can reverse aging and make us young again. This exciting research field instead focuses on specific functions that, in conjunction with other treatments and processes, could mitigate the effects and signs of aging. It is tempting to speculate that if this quality control mechanism works efficiently throughout life, can it ultimately lead to a healthier aging and extended lifespan? But the authors conclude, time will tell. And that ends my presentation. I hope you learned something about the role of cell competition in aging. Thank you and God bless us all. And here are my references.